Hi, welcome to the Physionic podcast. Today we're going to be talking about a series of different topics ranging from a lot on DNA, a lot on genetics, all the way up to if caffeine increases the risk of migraines, as well as variable heart rates. Well, before we get into that though, who am I? My name is Nicholas Verhoeven. I'm a PhD student in molecular medicine. I have my master's in exercise physiology, and I have been doing wet lab research for a number of years now in cellular biology and things of that nature. So, with that introduction out of the way, of course, as always, there's going to be timestamps for each one of the topics that I am going to be discussing in whatever platform that you end up consuming this. If that's on podcast or if you're actually watching the video format of this, then it will be linked for you as well. All right, so let's get into this. Before I actually get into this, I do want to mention something from a book that I'm going through right now. It's by Bill Bryson. It's called, I think, like a theory or an understanding of nearly everything in the universe or something along those lines. I'll have it written down somewhere so you can have access to it. But he talks about a series of different things, obviously, because it's all about different concepts in like geography, geology, physics. I mean, all kinds of stuff, history, biology, and really it's the biology aspect that I'm most interested in, and that's the part actually that I wanted to briefly discuss, because he mentioned something that I really hadn't considered before, but it makes some sense, and I'm curious on your opinion if you're listening to this. And he essentially makes the argument that our body is simply a vessel by which DNA is spread. And if you really think about it, I mean, every single one of our cells is filled with DNA to some capacity. And it seems strange to consider that DNA, little filaments or little strands of these repeating sequences of nucleotides, and I'll be going into that in a little bit, but these repeating sequences of, you can almost bring it down to a simplistic level of just four colors. You got blue, yellow, red, green, something along those lines. And the combination of those four colors, not mixed together, but literally just in sequential order, gives us our genes and gives us our, as you may have likely heard it, the blueprint of how we exist. And I mean, how our cells behave is largely determined by our genes, and what we look like is highly, highly dependent on our genes. And there's just so many things that go back to this, literally just these four identifiable units that are put in these random configurations, either like red, 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 blue, blue, yellow, blue, red. Like it's just like a series of different combinations of these colors, and I'm relating it to colors because I don't want to get into nucleotides specifically. But if you are familiar, if you've taken biology courses, what I'm actually referring to is not the colors, but referring to guanine, thymine, adenine, and cytosine. And with that, you get this creation of this structure that's just packed into this ball that's packed into this cell, which is essentially a ball in itself, depending on the shape type, the type of shape or the type of cell and its shape, its corresponding shape. And that is read over and over and over and over again to get different pieces of information to build different things. Just like an architect would look at their plans to be able to know, okay, this particular this is what it looks like on paper, and now we're going to make a 3D model of it and then put it into this house. So I think he has a point. I'm not going to necessarily distill our entire existence to we're just trying to procreate and we're just trying to expand our genetic line, but it seems so weird to me. It seems so weird to me to have these cells that are the smallest unit of life, and yet they are highly dependent on this non-living substance known as deoxyribonucleic acid, DNA. What a weird concept to think about. Anyways, I thought I'd mention that because it really struck me. And I am curious what your opinion is. If you feel like DNA really does have that strong of an input on our behavior and who we are. Anyway, that's just kind of an introduction into what I'm going to be talking about, which is a lot of DNA. Actually, I'm going to be talking a lot about some novel things that have happened related to research in the field of DNA. So to get into that research, I'm going to first explain to you a little bit more about DNA than what I've done, and I'm not going to simplify it to colors. Your DNA is found in an organelle known as the nucleus. The nucleus is, as I alluded to earlier, like a ball. And it has its own envelope. It has its own membrane. And that's what separates the inside of the nucleus from the outside of the nucleus. And the outside of the nucleus is called the cytosol. But that's still within the cell. All of that is still inside the cell. And DNA is kept up in these helical structures. I'm sure you've seen an image of what a DNA helix looks like, but it's got a bunch of what are called base pairs that are turned inwards, which is what the big discovery by Watson and Crick. They were the ones who essentially identified how these base pairs kind of match with one another, and that they're actually they're faced inwards as opposed to faced outwards, which was what a lot of other laboratories thought was happening. Some of them were using a tertiary structure, meaning that there were 3 strands creating a helix. I think you even had some single strand helices or some other structures. And Rosalind Franklin was the one who sort of figured out the actual structure as I believe she figured out that it was a double helix, but she definitely had an instrumental role in figuring out the structure of DNA. But Watson and Crick were the ones that actually found these base pairs. That's a bit of a tangent, but there's a little history lesson. So these base pairs are these pairings. You have, again, these 2 strands. So you can think of it as like 2 ropes that have a bunch of beads on them. 2 strings, there we go. 2 strings with a bunch of beads on them. And instead of using my color analogy, there's 4 different beads that you can put on these strings. And you can twist these strings together, and the beads will kind of connect with one another. And that's the base pairing that's occurring between the one base on one string and the other base on the other string as they're kind of twirled together. So that's that I mentioned, the guanine, adenine, thymine, cytosine, et cetera, et cetera. And those 4 units That's what I'm going to call them, beads, whatever you want to call them. They're called nucleotides. And they're made up of a series of different molecules. I'm not going to go into that, but they create life, essentially. They are inanimate. They're not alive themselves, but they hold the information of all of life through just their arrangement of one another and how they're related to one another, which is an incredibly simplistic way of structuring mass amounts of information. Think of like Morse code. It's incredibly simplistic, but it works. It does do the communication job. And I think the reason most likely for that is because you want something that is incredibly simplistic so that you're not going to get as many errors. If you have something that's overly complex, especially in an already incredibly complex system like a cell, you can't have a bunch of things that could go wrong. So anyways, you have these strings that make up this helix, and it's made up of all of these beads, which are synonymous in real life to these nucleotides or these bases. So that is the structure of DNA. Now, when our body, our cells, let's go all the way down to the cellular level, when our cells need to read something, your body, if you need to read something, you'll just go over to your bookshelf and take something off of your bookshelf and open it and read it. You can't read through the title page or the front of the book. You have to actually open it. Well, the exact same is true for your cells. They can't just look at DNA and be able to read it. They have to open it. So that's exactly what they do. They will go in, and there's a variety of different enzymes, different proteins that will go into the nucleus and will go to specific sites on DNA, and they will unzip this helix because, again, it's base paired. They're all paired together. It's not like you have this single string. You have two strings. But if you only want to get information off of one of the strings, you have to unzip them so that you can so you can attach to that string and read it. That's what your cells do. So they unzip it and they go about reading it, which then allows them to, and this is through what's called a polymerase. So a polymerase will allow the reading of the DNA, which will then produce a RNA. So a ribonucleic acid as opposed to a deoxyribonucleic acid. And then that gets translated. It's like your cells are translating it to different languages, but it's transcribing the DNA to RNA, and then it's translating this RNA sequence after more processing and stuff, which I'm not going to go into right now. It will translate that into a protein that is then functional, that can actually do things for the cell. So I'm telling you all this because it's important for us to understand the research that's being done around this topic. Now, I'm giving you a base understanding of how DNA works and how we read it and how our cells read it and things of that nature. But really what I'm trying to get at is what happens when your DNA breaks, which can happen. And it happens relatively frequently. Fortunately for us, our cells are absolutely filled to the brim with all these repair machinery that will go in and fix DNA. Now, a lot of that DNA repair is mediated by if you have a single base or a single nucleotide that breaks off, or it's kind of uneven, it kind of juts out a little bit from this double helix, then you have particular enzymes that will go up to it and attach to it, and it's called single base or excision repair. They'll flip out the base and either remove it or repair it and then flip it back into position where it needs to be. That's if you have a small problem, a small break in that double helix on one of the strings, on one of the strands. But if you have more extreme damage to your DNA, let's say you walk through a radiation field, not advised. The reason for that is because radiation will go through your DNA and wreak havoc on your DNA, which can potentially cause a series of different mutations. Again, not the topic of this podcast, but my point being that if you were to cause serious damage to your DNA, where long chunks of DNA, not just one base, but let's say 10,000 bases are all damaged, well, you can't have one enzyme that goes in and flips each one of those. That's incredibly tedious, and your cells don't have time for that. So what do? They use another enzyme called a nuclease. So a nuclease, there are endonucleases and there are exonucleases, but generally a nuclease will go in, find the point at which the damage has started to occur, and find the end point of where damage has stopped occurring, and will cut that entire segment of DNA damage and remove it. So then it's removed and it's free floating. And that's what the research is about. But before I explain what that research tells you, I'm going to explain to you a little bit about what we currently know about how DNA is repaired from that point on. So from that point on, the nucleases have done their job. You have what's called a DNA polymerase, which is, again, the same, a little bit like the polymerase that transcribes DNA to RNA, but this time a DNA polymerase will read the DNA from an intact DNA strand and replace all the bases, all 10,000 bases, all the way down that line, because at this point you only have one strand that's intact, and this is assuming that it's only affecting one strand. Most likely it's going to end up affecting both strands because they're in such close proximity with one another, creating that double helix. But if it only damages that one strand, then the DNA polymerase will come in, open that up, kind of separate out the two strands, and start start inputting single bases into that 10,000 nucleotide chunk that's now missing, and refill those 10,000 nucleotides, then close it back up. And now you have these other enzymes that come in called ligases, and they will literally, as the word implies, ligate, meaning they close, they solder in a manner of speaking the 2 points of breakage so that they get reintegrated into this double helix, and then you have an intact double helix which has been fixed from its radiation damage in our example, although this can occur due to a number of different reasons. So that's all fine. And there are more extensive methods of repairing DNA, like if you have double-stranded DNA breakage, which then requires a system called non-homologous or homologous recombination, but I'm not going to go into that. What I'd like to go back to is this DNA strand that's been plucked out with all of its mutations, right? All of its damage that's been occurred to it. What happens to it? Well, this is where the research comes in. This is where the recent research comes in. It's really, really cool. And it only gives us a partial look at what actually happens, but it gives us a really cool idea of what's happening. So this free-floating DNA can't just sit and free float. You can't just cut something up in the nucleus and then just, okay, well, we're done. The cells can't afford to not recycle, for one. And when it comes to proteins, they recycle that stuff to no end. Just continuously, as efficiently as possible, try and recycle as much as they can. Now, with DNA, the story is not quite clear. But what we do know is, again, they can't just have a ton of free-floating DNA that's just floating around in the nucleus. Eventually that starts to pile up. It's like trash on the side of the highway. You can't just have that stuff pile up, pile up, pile up. You have to have something that comes in and does something with it. And that's where this research came in. So there are what are called Rad proteins. And these Rad proteins will pair with the broken DNA, so this strand of free-floating DNA. And once it's attached to it, it will create a droplet around the broken DNA piece and the Rad proteins. And then this droplet will be attached to by, you may have seen this really, really cool video of this little protein kind of walking along a rope-like structure. It was a really famous video, maybe like 5 to 8 years ago. And everybody was, I don't know, they completely mischaracterized what it was. But it's a dynein protein or a kinesin protein. Now, these two proteins are critical for moving things across the cell. So from the edge of the cell to inside the cell to the right of the cell to the left of the cell. I mean, to every single area of the cell, it needs to be able to move things. The cells can't just have things just free floating just everywhere. It needs direction. And believe it or not, your cells have highway systems within them so that you can get direction, so you can be, okay, we need to move this piece of equipment or this protein to this location and that to another location, et cetera, et cetera. So that's why you have these two main protein complexes called dynein and kinesin. And these proteins are like taxi drivers or like Uber drivers or Lyft drivers or your mom. I don't know. Someone that's driving this system, UPS drivers, FedEx drivers, whatever you want. Let's call it UPS. So they latch onto this droplet, and they latch onto really anything, any vesicle, any sort of circular structure with a bunch of stuff in it, and will transport it to another location. Now, what's really, really cool about this is that they attach to this specific droplet with the broken DNA strand, and they have these Rad proteins that are attached to this DNA strand, and they fuse together several of these droplets together, several DNA strands that are broken, creating a much larger droplet, and they do this using suction. So they're able to create suction between the droplets so that they fuse together and you get this much larger droplet. So, and I've got this written down here in front of me as, I call it the DNA pop-up shop. And the reason why I'm calling it that is because these things create this massive droplet, massive, relative to like the nucleus, within the nucleus, on the periphery of the nucleus, because again, it's the envelope, right? It's a circular structure. So it's right on the edge of the nucleus, so it's kind of out of the way. And then these proteins will start working on these broken pieces of DNA and start fixing them, start repairing them. And that's what the research is about, that it's essentially these pop-up shops that start to pop up inside the nucleus, and they just start tinkering away at these broken strands of DNA. Now, the mystery, that's so cool. That is so cool. But what we don't know yet, or at least this paper didn't look into it, but and I don't know, I haven't read anything on this, but as far as I know, we don't know what happens to these pieces of DNA. Are they completely fixed? Are they attempted to be fixed? and then they get destroyed. We're not sure what happens. Well, I guess I shouldn't say we, as in the scientific community. I'm not sure what happens. If anybody knows, feel free to send me a paper and I will read it. So, but up to that point, that is a really, really cool concept, to think that your DNA is taken care of in these droplets, and then you make these pop-up shops where you have these DNA machinery pieces that kind of come in and start fixing and doing whatever they need to do to make it viable for something. Maybe it's reintegrated somewhere. I don't know. Maybe it's, I genuinely don't know. I can't, maybe in certain situations you don't have the DNA polymerase come in and fill in each section. You just wait for this droplet to finish its repair process, and then you bring it back to that section of DNA and insert it back in and ligase both ends, and voila, you're done. I don't know. I don't know the answer, but that still, up to now, what we know is ridiculously cool. That is ridiculously cool. Keep in mind, cells are the smallest unit of life, and they don't have a brain. They're not thinking about this stuff. It just happens. Remarkable. Okay, so I've been railing about this for 26 minutes now. Let me talk about the two other topics that I wanted to discuss. One is a study that looked at variable heartbeat. So you may be familiar with the idea that your heart rate is, set at a certain point. Let's say like the average heart rate, I don't know it, I'm just throwing out a number. Let's say it's 50. And people sometimes, and certainly exercise physiologists, look at heart rate because it tells you some information on how the heart's functioning and things of that nature, but maybe we should be taking a step back from that. I think we can still look at trends based off of the research I'm about to explain, but we may not want to look at the exact heart rate and just make a ton of assumptions based off of that heart rate. So let's say the average heart rate is 50. The thing is that this study investigated for up to almost a year's worth of heart rate data from 92,000, almost 92,457 individuals, and they wanted to figure out what was kind of the average heart rate for all these individuals over almost a year's worth of time. And what they found after collecting all of this data is that there's substantial variability between individuals, meaning that for some individuals, their heart rate might be 65, some people might be 50, some people might be up to like 100 at resting levels. And not all of it can be explained by saying, oh, they're unhealthy if they have a high heart rate. Typically, the trend is that if you have a lower heart rate, you tend to be healthier, just because then you have a stronger heart that's pumping more blood per beat. Therefore, you need less beats. That's the idea. And for a high exercising individual, yes, that's almost universally going to be true. For the average individual, that may be a little bit more in question now. And for individuals that may have a heart rate that's slightly higher, that may not be as indicative as we once thought in regard to if a person is healthy or not. And of course, it matters if you're feeling anxious that day. I mean, the problem with the study, of course, is that they're just looking at this one dependent variable. They're just looking at heart rate. They're not taking into consideration literally anything. So these people could be high functioning anxiety individuals for some of them, which of course they've got a sample size of 92,000. So this is all going to wash out. But the idea being that there's still going to be immense variability. So you can have some people that are way up in terms of their heart rate, like 90 at rest. I don't know if that's actually the case, but I'm just saying, like, it could be way up there because they did mention a variability. as extreme as 70 beats per minute or more or less than another person's. So if the average is like 50, you could have a person that's like at 110 or 120. And that's going to be dependent on exercise, your fitness level, what you're eating, if you've taken caffeine, how much sleep you've got. I mean, just go down a laundry list of different reasons why people's heart rate might be so variable. It could be any number of things. But the main takeaway here is that we can probably still follow trends that a lower heart rate tends to be better, especially, and you can be for sure that's the case if you exercise quite a lot, especially aerobically exercise, cardiovascular exercise, long duration exercise, not weightlifting, talking about running, biking, rowing, things like that. But that does not necessarily mean that just because you have a high heart rate, like if you are generally healthy, fit, and you have a heart rate that's like a little bit higher than average, that doesn't necessarily mean that you're in an unhealthy situation. That's essentially the point that I'm trying to get across. And I think that's the point that this study was trying to get across. So heart rate variability is quite large, and heart rate data is going to be all over the place. A lot of variance, to use a statistical term. And finally, the last topic that I wanted to discuss is caffeine cause of migraines. Does caffeine cause migraines? Well, we need a lot more research on migraine research. I've talked about it a little bit in the past in previous episodes, but caffeine, according to this study, which was an associative study, it was a prospective associative study, meaning that they're not confirming, they're not controlling for a ton of variables and then giving people caffeine and then seeing what happens. They're not doing that. They had 96 or 98 individuals that were identified as frequent migraine sufferers, and they were told to record their daily food intake and their beverage intake for 6 weeks in the morning and in the evening. The morning was probably more just, did you suffer a migraine? How do you feel? Things like that. But in the evening, they had to record what they'd consumed and if they were experiencing a migraine within that day. And I'm sure probably the severity and things like that. So then the researchers wanted to standardize how much caffeine these individuals were consuming. And I imagine that they probably did not tell these individuals, hey, we're measuring how much caffeine you're intaking, because that would be an unnecessary variable that they're throwing in there. But I don't know if they did that or not. Regardless, I digress. So they standardized the amount of caffeine that people consumed in terms of what they interpreted as a standard amount of caffeine. So a 12-ounce can of soda, 8 ounces of coffee, 6 ounces of tea, 2 ounces of energy drink were all considered equal, the same. And this will, of course, still give substantial variability because no single cup of coffee is going to be the exact same as another cup of coffee. So that's certainly the case. You can't necessarily compare between subjects, between one person and another person, but you can compare a person at the same person at 6 weeks before and 6 weeks later, or throughout the study after 3 weeks and, after another 3 weeks, because, habit is there. And that's something I've spoken on, a number of years ago. I spoke on this like 4 or 5 years ago, that maybe because we're such habitual creatures, we're such habitual animals, we're humans, that sometimes it's a moot point to say that if you consume, to sit there and try and control for every single variable, especially when it comes to nutrition, because people typically stick to the things that they like and they stay away from the things that they don't like, and they've already figured all that stuff out. So they're most likely over those 6 weeks going to continue consuming what they've consumed. if you drink coffee in the morning, it's highly unlikely that suddenly you're going to stop drinking coffee over those 6 weeks, or after, you have one day where you consume coffee, another day where you consume tea. You drive 3 miles one way to a tea shop to get tea. Then the next week you drive 16 miles to go get coffee from this particular Starbucks. We don't have that much variability in our habits. That's my big point. So while the differences between the subjects, a person in New Jersey compared to another person in New Jersey, they live, 2 miles away from one another, and they go to different Starbucks, and those Starbucks have different amounts of caffeine within each Starbucks coffee, even though they're buying the exact same Starbucks coffee, the venti, macchiato, supremo, whatever. I don't know Starbucks very well, clearly, but there may be variability, so it's hard to compare between those two individuals, but they will continue to go to those Starbuckses. That seems a little off. Those Starbucks, and because they will continue to go to those for the next 6 weeks, there's going to be less variability when you compare them to themselves. That's my big point here. So anyway, that was a long way to get to my overall point. So what did the study find? Let's just touch on that. They found that consuming 1 or 2 caffeinated drinks based off of the metric that I gave you earlier, 12 ounces of soda, et cetera, did not have an association with increased migraines. However, having 3 or more caffeinated drinks did have an association with increases in migraines. So if this is causative, as always, this is an associative study. We can't say. However, at least it gives some benchmark, some information on how caffeine is associated with migraines. And of course, the type of migraine that a person suffers. Is it episodic? Which in these individuals, it was. I believe all these individuals had episodic migraines specifically. So this is some evidence that if you consume some caffeine, you shouldn't necessarily be afraid that is the reason why it's causing migraines. Now, of course, they would actually need to do some controlled studies to really hammer that down and make sure that's the case. But at least preliminarily, it gives us some confidence. Clearly, I'm very confident here. Some degree of confidence that a small amount of caffeine, 1 to 2 drinks of caffeine, will be fine. You won't suddenly see your migraine pop up again. But having much more than that may increase your prevalence of migraine. So try and keep caffeine to a small amount so that that won't increase the prevalence of caffeine compared to individuals who consume no caffeine. So no caffeine to 2 cups of caffeine, fine. 3 cups or higher, probably not fine based off of the data presented here. Whew, man, I made that a little bit longer than it probably needed to be. Anyway, that's what I've got for you. Hopefully you found this informative. And with that said, I wish you a wonderful, wonderful day. And I hope I have the absolute pleasure of speaking with you in the next one. I will talk with you guys next week. Have a good one, guys. See ya.